Hello. Welcome, everyone, to the first of this summer's Wednesdays at the Genome. I'm very excited that everyone's here to participate today. This has been a tradition for the Department of Genome Sciences for at least the last decade. I probably should have figured out how long it's been going on for. These are really some of the most fun talks that we put together for the entire year, which is really geared towards a more general audience, trying to get people to know more about genomics, as well as sort of what the cutting edge research is going on right now, as well as where the field has been in the past and how this relates to everyone's normal life out there. I'm really excited to introduce Bob Waterston, who is the founding chair of the Department of Genome Sciences. Bob really has been a founder, not just in the department here locally, but in the field broadly of genomics, playing a leading role in the Human Genome Project, the Mouse Genome Project, the Nematode Genome Project, which I'm sure you'll talk a little bit more about that for those who don't know what a nematode is. You'll probably explain that. And in addition to all of this stuff, he's received numerous awards for this. He's a member of the National Academy, received a Gardner Award, amongst others, which I don't really have time to list them all because I want you to actually talk, not me. But we're really excited for you to be kicking this off, and also recognizing the fact that, in light of the many illustrious stuff he's done throughout his career, he just retired like within the last 2 weeks. So this is more of a talk about as well of what he's done and sort of the beautiful work that really he's led throughout our field. And really without him, we would be in a very different spot in terms of genomics right now. So thank you very much for leading us off this summer. I really appreciate it.
Thanks, Andrew. It's on now, right? Okay, can you hear me all right? Good. Okay, we're going to start off with a little clip here from a little while ago.
Nearly 2 centuries ago, in this room, on this floor, Thomas Jefferson and a trusted aide spread out a magnificent map, a map Jefferson had long prayed he would get to see in his lifetime. The aide was Meriwether Lewis, and the map was the product of his courageous expedition across the American frontier all the way to the Pacific. It was a map that defined the contours and forever expanded the frontiers of our continent and our imagination. Today, the world is joining us here in the East Room to behold a map of even greater significance. We are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind. It was not even 50 years ago that a young Englishman named Crick and a brash, even younger American named Watson first discovered the elegant structure of our genetic code. Dr. Watson, the way you announced your discovery in the journal Nature was one of the great understatements of all time. This structure has novel features which are of considerable biological interest. Thank you, sir. Thank you. How far we have come since that day. In the intervening years, we have pooled the combined wisdom of biology, chemistry, physics, engineering, mathematics, and computer science, tapped the great strengths and insights of the public and private sectors. More than 1,000 researchers across 6 nations have revealed nearly all 3 billion letters of our miraculous genetic code. I congratulate all of you on this stunning and humbling achievement. Today's announcement represents more than just an epoch-making triumph of science and reason. After all, when Galileo discovered he could use the tools of mathematics and mechanics to understand the motion of celestial bodies, he felt, in the words of one eminent researcher, that he had learned the language in which God created the universe. Today, we are learning the language in which God created life.
Okay. So it's not often that science gets announced in the White House by the president, and indeed with such lavish praise. Tony Blair, the UK Prime Minister, was also linked in by video with a similar press conference going on in London. He was highlighting the Anglo-American collaboration part of it. Did you spot me in the audience? Okay, and my wife, Pat. We were there because I was one of the leaders of the groups that sequenced the human genome. And today, I wanted to tell you how I ended up in that audience and a little bit about my perspective about how that achievement came to be. The structure that Clinton referred to is the double helix with A paired with T and G paired with C. It's the order of those four letters, as indicated on the right, that spell out whether we are a chimpanzee or a human or have black hair or blonde. The genome of an organism is all these letters. It's all its genes, its genetic code. In the case of viruses, it might have only a few thousand bases, a few thousand letters. For simple bacteria, it might be a few million. For human, it's 3 billion base pairs or letters. 75,000 pages of the New York Times. Our task was to find out each of those letters in order, spell it all out, what our code is. So to begin, I take you back to, there's going to be three parts to my talk. There's going to be the beginnings, the people who influenced me, got me into the field, the frying pan, and the fire. So to begin, I take you back to 1969 to Woods Hole, Massachusetts. Sidney Brenner was strutting across the stage, talking for the first time in public lecture about a new project he was undertaking to study a tiny nematode, Caenorhabditis elegans, a name longer than the worm. And that's that worm you see on the right. Sidney was well known already for studying the basics of DNA in bacteria and their viruses. Now, he was proposing to use genetics to learn how DNA managed to specify the development of an animal and its nervous system with all its associated behaviors. Oops, we don't need that. The worm is tiny, just a millimeter long, with only 959 cells, 302 neurons, yet with quite sophisticated behavior. You could see it crawling across and doing lots of things. Its small size means that you can grow millions in the laboratory, and that's important for genetic analysis, where you're often looking for very rare events. He showed pictures of the mutants, electron micrographs of the nervous system, and he laid out just how he planned to attack the problem. Sidney was a real showman. I wish I could emulate him. He had the audience in the palm of his hand. I was in that audience as a student in the physiology course at the Marine Biological Lab. I began medical school in 1966, fully intending to become a practicing physician. But in my medical school courses, I became increasingly interested in understanding the basis for disease, believing that a greater understanding of what led to the disease would lead to improved therapies and treatments. Frank Fitch, a professor in the pathology department, enticed me to join his lab to study the cellular basis of the immune response. And I love the puzzle of it all. But as I took more courses and learned more biology, I was an engineer as an undergrad with no biology courses. I became more interested in the molecules within the cells. And the physiology course I saw as a chance to learn more about that arena. I was only supposed to attend the first half of the course, the first half of the summer. But a couple weeks into the summer, or into the course, Andrew St. Georgie, the course director, persuaded me to stay for the second half so I could meet a man called Sidney Brenner. Sidney was going to lead a noonday session every day, a literature review, and he would talk about whatever we asked him to talk about the day before. It was astounding. He had incredible knowledge. By the time of Sidney's evening lecture, I was already in his thrall. By the end of the course, I decided I wanted to do a postdoc with him. I approached him on the beach. He looked at me askance and said I should write to him when I got home. I thought it was a polite refusal. But to my amazement, when I wrote, he accepted me, and thus began my work on the worm C. elegans. I worked on genes involved in muscle. It was a steep learning curve, but I delighted in the geneticist's approach of taking out apart a gene and seeing what went wrong, trying to figure out what that gene was doing. We found lots of mutants and published a couple papers. And with that, I was able to get a job at Washington University, not University of Washington, and in the anatomy department. So that's worms. That's the beginning in worms. In yeah, you had that, right? Yeah, okay. Now it's not gonna go. What'd I do? What'd I do, Justin? It doesn't want to go. What? Let's try that. There we go. Now it's live. Okay. All right, now we're up to 1978. I was on the faculty search committee for the nascent Department of Genetics. I had lunch with a candidate, Maynard Olson. Maynard had given his job seminar on his work with Ben Hall here at the UW, in where he used DNA polymorphisms as a genetic trait to put genes on chromosomes. While that would form part of his research program, that wasn't what he wanted to talk about at lunch. Instead, he wanted to describe his plans to map the entire yeast genome. Maynard's idea is shown on the right. He would take DNA from yeast cells, fragment the whole genome, fragment it into smaller pieces, and insert the individual pieces into the bacterium E. coli. Essentially purified each piece away from the mix in a process that we call cloning. And if you look here at the top, this is some chromosomal segment. Here we have some random bits that have, these are the fragments, the smaller fragments that are broken. And by the way, this is a Leroy lettering set diagram that Maynard wrote in 1979, I think. It still works. So these are the different fragments. The vertical ticks are sites that a restriction enzyme recognizes. You can think of a restriction enzyme as sort of like a search function in a Word document where it searches out a particular set of letters. And in this case, the restriction enzyme cuts at these particular sites, producing fragments of different lengths. And you can measure them as shown down here where the largest is at the top, the smallest is at the bottom. And clones that overlap share fragments. So these all have the same fragment, and these have the same fragment, and so forth. And by that process, you can determine the overlap and reconstruct the whole. So you can take, no one of these clones spans the whole thing, but you can get a representation of that. If you take thousands of those clones and use a computer to recognize overlaps, Maynard's idea was that you could reconstruct the whole yeast genome, not just this little bit, but the whole 13 megabases of the yeast genome, 13 million bases. I thought he was crazy at first, but then as I listened to him and how carefully he had thought everything through, I realized that he could do it. He might be able to do it anyway. And the result, sort of an encyclopedia of the whole genome, where you could just reach up and grab whatever volume you wanted, access whatever gene you wanted, was an astounding idea. And to me, that was the birth of genome science. That's the first I heard of it. Maynard came to WashU, and I finagled my way into the genetics department, into the lab next door to his. We became fast friends. I was on a hike with him on that mountain that we saw at the beginning. I watched him build the yeast map with fascination and envy. But the worm genome at some 8 times the size of the yeast genome seemed beyond reach. Besides, I had a thriving lab studying muscle. Okay, next. In 1985, I went on sabbatical back to Cambridge to study the embryology of muscle. As luck would have it, the only available space in the whole building was in John Sulston's lab. John had earlier figured out, let's see, the lineage of C. elegans, where you go from the one-celled egg to the 959 cells of the adult, all the different divisions. He mapped all those out, and that won him a Nobel Prize in 2002. But John was not working on the lineage anymore. He had started in 1982, he'd started constructing a clone map of the worm genome, analogous to what Maynard was doing in yeast. But as far as I know, he came to the idea independently. And he was using larger fragments of DNA and a different method of analyzing to account for the larger worm genome. But the overall strategy was essentially the same. By the time I arrived on sabbatical, John, along with Alan Coulson, had analyzed some 7,000 clones. With that many clones, they should have had 95% of the whole genome already represented, but it only was 80%. And the gaps in the map weren't closing at the rate that you would expect. This result exposes a vulnerability in this random approach. If the set of clones isn't really random, if there are problems in representing different regions of the genome, there'll be holes in the map. And the idea was that certain sequences were just not well tolerated in E. coli. And during that year, sitting in the lab with John and Alan, I became drawn into trying to find a solution. The answer came on a visit back to St. Louis to check up on my lab. Maynard's lab had been developing methods to take large pieces of DNA and put them into yeast instead of E. coli. And yeast, as a eukaryote instead of a bacteria, would be expected to have different biases. I returned to Cambridge excited. John thought it was worth exploring. And when I got back to St. Louis, I started putting DNA into yeast. I shared the clones with John, and with encouraging preliminary results, we moved ahead full force. And in a couple years, we closed all the gaps, basically. And it was also the start of a collaboration that would go on for some 15 years or more. This nearly complete map was the ideal substrate for sequencing the worm genome. But given time limits today, I won't be able to tell you how John and I ended up sequencing the worm genome, our adventures there, the founding of the the founding of the Sanger Center by the Wellcome Trust, the firing of Jim Watson from the NIH, and other good stuff. In short, with Jim Watson's okay and funding, we began sequencing the 100 million base genome of the worm. Even though at the time, the world's repository for all DNA sequence had a measly 20 million bases from all sources. We were going to do 100. Anyway, working together as one lab with rooms 4,000 miles apart, we had success. People noticed. We ended up on the cover of the New Statesman, celebrating our transatlantic collaboration, and on the cover of the Cold Spring Harbor abstract book with John and me well out in front of the other groups with Watson up there. Rick Wilson, the head of sequencing at WashU, had been asked after a talk at a meeting what he felt was the biggest challenge in scaling up like this. He paused only for a moment and replied, lab romance. Lab romance. And it was true. We had hired young kids out of college, and they were soon dating. The dating wasn't the problem. It was when they broke up that we had difficulty. Okay, so that's beginnings. Into the frying pan. John and I were sipping sherry in his garden it was September 1994, the last day of our annual joint lab meeting. This isn't really us in the garden, it's from another shot, but it's a nice picture. Things were going very well for us. Our paper reporting 2.2 million bases of contiguous high quality sequence had come out. And with a variety of advances, including more automation, that were already available or on the horizon, we thought we could speed things up even more. Phil Green had developed his base caller that gave statistically rigorous error rates. And using that, he could do a much better assembly of the sequence. In any case, this touchpad is too sensitive. Anyway, as with the mapping project, we were doubling our throughput every year. And the prospects of actually completing the worm genome by 1998 seemed better than ever. Yet John was quiet, almost sullen. When I pressed to find out what was bothering him, he said he was feeling pressure from his funder, the Wellcome Trust, to do more of the human genome, and he didn't know how he was going to manage that. So on the long flight back to St. Louis the next day, I began to ponder just what would be needed to make a reasonable assault on the human genome. It gradually dawned on me with the advances that we had already made and the ones that we could foresee, sequencing the human genome was no longer a preposterous goal. So you can understand my ideas. I have to take a minute and explain to the sequencing strategy we were using. We took a clone from the map, say that big, broke it up into smaller pieces, and then got sequence from one end of that small piece. These were random bits, much like the random bits that we used in the map. But this time getting sequence. And by comparing the sequences, just like you could compare fragments, you can detect overlaps and thereby get bigger and bigger pieces. This is a process called shotgunning. It's named after the scatter shot of a shotgun. It's not directed, you just blast things. That, just like in the worm map, that works pretty well, but you almost always are left with gaps and regions of lower quality. And you have to go and fix these. So in addition to the shotgun and its assembly into bigger parts, you have a finishing or polishing phase. And what I realized was that the advances were coming more rapidly in the technologies that we used for shotgunning than they were for finishing. And so if we concentrated on the shotgun first and did finishing later, we could get most of the sequence much faster. In fact, my projections were that we could get the sequence by 2001 instead of the 2005 goal that had been the official. That was the endpoint in the official stuff. And by concentrating on the finishing afterwards, giving us more time to get better at it, we could get the whole thing in high quality, no gaps, or at least minimal number of gaps by 2003, 2 years ahead of schedule. I worked through the details of what could be done. And by the time we landed, I had a plan. I was punching out the numbers on my calculator watch. I show a picture because probably half of the audience doesn't know what a calculator watch is. You have laptops or whatever, or phones. Anyway, I calculated that we would only need 50 million fragments. Only 50 million. Anyway, after sleeping on it, I decided it wasn't just hypoxia-induced mania. I emailed John, spelling out all the details. For mostly political purposes, I said that our two groups would only sequence 2/3 of the genome, leaving the chance for other groups, particularly international collaborators, to participate and contribute. John initially was incredulous, of course, but as he digested it, he decided it wasn't crazy. And as John and I shared it with other members of our groups, the excitement grew. Soon we had dubbed it the Megalomaniac Genome Project, or MGP for short. We indeed had a plan for getting the human genome sequenced. I knew one of the most dubious people would be that Maynard Olson, who by that point had moved to UW. Maynard was firmly in the camp of feeling that the current technology was not up to the task of sequencing the genome, and some new technology would be needed. I sent him a draft of what our proposal and followed up with a phone call, and Maynard was cautiously on board. To go forward, we would actually have to have money. We would have to persuade our funders that this was a good idea. John arranged a meeting for early December to present his plan to the Wellcome Trust. I sent a draft proposal to Francis Collins, now the head of what was eventually called the NHGRI, in early November. He asked me to present our ideas to an already scheduled meeting in mid-December, shortly after the Wellcome meeting. And he arranged a meeting with Harold Varmus, now the head of the NIH. We were being taken seriously, and we even ended up being our ideas published in Science as a News and Views article. So far so good. But on the Saturday before the Wellcome meeting, disaster struck. Daphne, John's wife, called me to tell me that John, while riding his motorbike to work, had been blindsided by a car. He was unconscious with a shattered pelvis. and in the operating room. My world was upside down. The next day, I spoke to John, who was out of surgery and had regained consciousness. He insisted that our meeting scheduled for Thursday should go ahead and that I should come as planned. Nuts. But I agreed to come. I went the next day, I think, mostly to see for myself how he was doing. When I visited him in hospital, he was doing very well, almost euphoric. Maybe the painkillers, I don't know. But he was insistent that the meeting go on, and he finally persuaded his doctor to let him go by telling him that I was a physician. Never mind. Never mind that I hadn't seen a patient in 20 years. So on Thursday, I wheeled John down the hall, out to the limo that the welcome had arranged. And John was amazing at the welcome. Again, maybe the painkillers, I don't know. But the welcome people were clearly enthusiastic. They thought this was going to fill their mostly empty Sanger Center that they had just invested in. I met with Harold Varmus the next week. And the following day, I took the podium at the Mapping and Sequencing Center Directors Meeting. That's the centers across the country who were doing mapping and sequencing. And most thought I was going to present an update on the worm sequencing. Instead, I presented our plans for the MGP using a series of overheads. Again, you don't know what overheads are, but they were there. The audience was very quiet as I finished, until Maynard stood up and said to the group that they had before them the first credible plan to sequence the human genome. In the discussion that followed, there was gratifyingly little skepticism that we could deliver the sequence at the rate we proposed. There were some questions, but most were readily dealt with. However, the biggest question centered around the absence of a clone-based map, the kind that had fueled the worm project. But overall, I thought it went about as well as I could have hoped. Then the maneuvering began. A major player here was Eric Lander, a most formidable fellow. These are some of his notable past accomplishments. He led a highly productive team at MIT, and he prided himself on recognizing when a field was ripe for scaling up, and he focused on automation to make that happen. He had done this very effectively on the mouse genetic map and was now doing it on a different kind of map of the human genome. It was widely held that in a couple of years, he would likely launch an effort to sequence the human genome himself. Our proposal caught him on his back foot. He needed time to transition his group. He characterized our efforts as cottage industry, arguing that he was prepared to provide the automation that was needed to do the job right. He conveniently ignored the fact that with our cottage industry approach, over the past 4 years, we had already scaled up sequencing 30 to 50-fold, and we dropped costs 5 to 10-fold. We needed just an additional 10 to 20-fold to achieve our objectives. And with the advances on the horizon, we were sure we could do that. Despite his publicly stated misgivings about moving ahead, he began to talk with John and me about collaborating. We hosted his people, freely showing them everything we were doing. We even sent formal letters of collaboration. At the same time, he and I were competitors. We submitted competing grants to the NIH to do more sequencing. And by extension, he was a competitor of John's. So we were surprised when he was appointed an observer on the review committee That was to consider John's formal proposal to the Wellcome Trust. Rather than a quiet observer, Eric aggressively challenged John's projections using a calculator with large buttons, punching in numbers and casting out doubt. John was not expecting this kind of grilling. It's not very English. It even seemed to take some of the other committee members aback. I was in the audience, but I was forbidden to say anything. In the end, John was awarded only about half what he requested. On the other side of the Atlantic, Francis Collins took a cautious approach. Ignoring Andrew Carnegie's advice, put all your eggs in one basket and watch that basket. Francis opted for a program that funded multiple labs at modest levels over 3 years. The rationale was to allow labs with different ideas to compete before committing to a more expansive effort after this pilot phase. Notably, there was no program to develop a clone-based map of the human genome, of the kind that was driving the worm sequencing. The result was that on both sides of the Atlantic, our collaboration was substantially slowed from what we thought we could do. During the pilot phase, no alternatives emerged to the methods and strategies that we were using. The groups making the most progress were doing more or less the same things we were. But beyond this coalescence of how to sequence, there were some real benefits. Through gatherings of the lab heads and their funders at Bermuda, we developed means of data sharing, of figuring out who was going to sequence what and collaborating. And in addition, there was also the more renowned principles of rapid data release and no patenting, known as the Bermuda Principles, but I'll come back to that at the end. With the consolidations of the groups around a single strategy and a way to share things and means to collaborate, by the end of the pilot project, even Francis had to recognize that it was time to scale up and tackle the whole genome. While all this was going on with the human genome, our labs continued to beaver away at the worm genome, and we helped get the yeast sequence out the door in 1996. We continued to post sequence on the internet as we got it, and we were gratified to see its wide acceptance. Postdocs and grad students no longer spent tedious hours cloning and sequencing their favorite gene, but could instead get to work on exactly what that gene was doing in the animal. With only the most challenging bits left in 1998, we published the worm sequence, 97 megabases of it, with lots of fanfare, including joint press conferences in London and Washington. Harold Varmus and Bruce Elberts, the president of the National Academy, sang our praises. And Bob Horvitz, who had urged us in the beginning to start sequencing the worm, said it was better than walking on the moon. It was the first animal genome sequenced and the first genome of a size even comparable to anything else like the human. It was what we had begun this whole journey for. And with this milestone, John announced that he was stepping down from the leadership of the Sanger Center. He would continue to be involved, but would not have the responsibilities of director. Okay, into the fire. In the spring of 1998, nearing the end of the human genome pilot phase, we were attending the annual meeting of the Genome Crowd at Cold Spring Harbor. Michael Morgan, the staff person at the Wellcome Trust handling the Sanger account, had just finished an unscheduled talk when he received a standing ovation. A staff person, a standing ovation. What was going on? Well, the prior week had been a mess. Craig Venter announced the weekend before through a press release and an exclusive interview with Nicholas Wade, the science reporter for the New York Times, that he would be forming a new company that would sequence the human genome much faster and much more cheaply than the public effort known as the Human Genome Project, HGP. The company was being formed by ABI, the instrument manufacturer that instruments everybody used, and its parent company, Perkin-Elmer. And ABI would make available to Craig, the yet unnamed company, 230 new generation capillary sequencers. And Craig would pursue a whole genome shotgun. Instead of doing it clone by clone, he would take the whole genome and shotgun that. It had previously been proposed by Jim Weber and Gene Myers. The business plan was a bit vague, but would involve some patenting of genes. And a careful reading of the press releases and other material suggested that despite assurances that the sequence would be available for free, as opposed to freely available, the new company sought hegemony over the genome sequence information and any analysis of that sequence, including derived discoveries. Since Venter would be doing the human, he suggested that the public effort switch to mouse, an obvious ploy to remove any competitors. It's not a good business plan to charge customers for information they could get elsewhere for free. Shockwaves spread through the HGP labs. The initial response from Collins and Varmus was not encouraging. They were all too welcoming, making it seem like the NIH might roll over and play dead. John, who had his own major expansion proposal already under consideration by the Wellcome, worried that the trust might simply say, if somebody else is going to do it, why should we pay for it? A thought that echoed across our side of the Atlantic. We knew that simply assembling shotgun reads from the whole genome wouldn't be any better than shotgun sequencing from individual clones, leaving gaps and areas of low quality. But there was no mention in any of the press releases or anything of any plan to take care of those gaps and make it really a high quality sequence. That didn't stop Venter from making vague assurances that the sequence would be of a quality equal to or better than the public effort. Before the Cold Spring Harbor meeting was going to start that Tuesday, the various groups of the public effort from across the world were already scheduled to meet to discuss progress and plans. Instead, we had Venter, who was actually part of that group, come and present his plans. Craig was downright insulting, and we were a hostile audience. It was not pretty. He reiterated his intent to patent just a few hundred genes and to release the sequence to the public on a quarterly basis without mentioning any conditions. At the same time, the business plan for this new company was to become the definitive source of genomic and associated medical information that will be used by scientists to deliver improved healthcare, the definitive source. Sounded to me like they wanted total control, not only of the sequence, but all the findings that might come from it. But the question was how to respond. We knew of the shortcomings of shotgun sequencing, as I said, and that going from a shotgun to a finished high quality sequence would be expensive and time consuming, especially with only whole genome shotgun data to go by. On the other hand, huge resources were being given to Venter, maybe as much as $1 billion. He was talented at manipulating the press with vague statements, half-truths, and selective omissions. He's very good at it. We were no longer simply dealing with scientists, reviewing scientists, but with the press and public opinion. With weight of the New York Times, Venter had already successfully portrayed us as old fogies, wedded to old technologies and afraid of the daring new approaches that he was taking. We would learn that reporters were not interested in learning about genome sequencing. They were interested in sound bites. While all this was going on at Cold Spring Harbor, John and his colleagues were presenting to the Wellcome Trust their plans to scale up to the full 1/3, their initial proposal. They made their prepared presentations, but John talked a bit at the end about the Solera proposal and pointed out that the Sanger was the only major center outside the US. Without their participation, the genome would be an American property. John and his colleagues left the room, but didn't have to wait long before the board enthusiastically endorsed their proposal, urging them to continue, especially in the light of the competition. It was in that context that Michael Morgan was given an unscheduled slot in the Friday morning session. Michael announced the commitment of the Wellcome to have their portion of the genome done and in the public domain. And in the excitement of the moment, he added that the Wellcome would pay for the whole genome if necessary, though he had no authority for that. That was what elicited the standing ovation. So the next 2 years, the next 2 years leading up to the White House announcement were really wild. There were many events and lots of stories from this period that I won't be able to cover here. But if you are interested, I recommend The Common Thread, a book by John Sulston and Georgina Ferry. I had a lot of input into that. My story's in there too. It's a good read. I want to focus though on two things for the rest of the talk. I'm going slow, so I apologize. I want to deal first with getting congressional support and the other with the physical map. While the Sanger Center was assured of funding for its assault on the genome, the US program was facing lobbying efforts by Solera to a Republican-based Congress, business-friendly Congress, to sidetrack the public project. A key congressional hearing was held in June 1998 with Venter and Collins as witnesses, along with Maynard Olson. Maynard made a couple key points. First, he stated, what we have here at the moment is neither new technology nor even a new scientific activity. What we have is a press release. Later, he went on to say he was prepared to be proven wrong, et cetera, but I'm comfortable predicting that there'll be over 1,000 serious gaps in that final product. A committee member, a congressman from Michigan with a PhD in physics, picked up on this quality issue. And in the Q&A that followed, he challenged Venter on quality. Craig's answers were, again, full of vague assurances, but Maynard knew he'd won the day when the congressman wrapped up his remarks saying, I find this very interesting. And as Dr. Collins observed, this is an experiment. It will be fun to get you back here in 3 or 4 years and read back your testimony. So he wanted the experiment to go forward. Maynard has written a very excellent piece on that shown at the bottom. It reads like a New Yorker article. It's really good. It's a really good read. Okay, with all this jockeying going on, the labs of the public effort were hashing out just how we could proceed. Over the next year, we gradually settled on a strategy where we would stick with our clone-based approach, but dissociate the shotgun from the finishing, much as we had proposed 3 years earlier. We would purchase the same capillary machines that Venter would have. The arms suppliers do well in any war. With those coming, we picked a level of shotgun coverage that we thought we could achieve by mid-2000. That was the time we figured Solera was going to make their announcement of victory. Finishing would continue, but would be a lower priority at that time, with a goal set of 2003 for a high quality sequence. Should sound familiar. The 3 US labs that had produced the most sequence in the pilot phase would join the Sanger in the DOE program, Department of Energy program, to do the bulk of the sequencing. Smaller US labs and international groups, including the French, Japanese, Germans, and the newly constituted Beijing Genome Center, would also contribute useful amounts. But the other major issue facing us was clone supply. It almost brought the whole project down. In the pilot phase, the NHGRI did not invest in the clone map, as I said. Instead, each group followed its own procedure, did things locally. It was not going to scale up. And Eric Lander was particularly unhappy with the clone-based approach. He saw it as a diversion of resources away from the shotgun effort, and he was loath to compromise. In the fall of 1998, Eric put forward the idea of sequencing random clones and using an initial bit of sequence from each of those clones to see if somebody else was already sequencing that and they should stop or go forward. Using such random clones would have been a major course change, disrupting how the whole project was organized. While I could see some advantages of the random clones in theory, I just couldn't see how it would actually work in the lab. I relayed the plan to John, who was not at this meeting, maybe deliberately so, and he was even more dismayed by the idea of the idea, and also by Francis's apparent willingness to push ahead with it. To John's further dismay, Francis had neglected to tell their funding partner, the Wellcome Trust, anything about his change of plans. By the end of October, John felt compelled to write a protest letter to Francis, warning him that he was in danger of destroying the international public program, something Celera had so far failed to do. The passion of John's letter finally clued Francis into the depth of disagreement of many others to Eric's proposal. Things finally got resolved in December 1998 in a hastily arranged meeting of the 5 largest centers. In the end, John and I agreed to build a genome-wide fingerprint map, a clone map, so for the whole community, and to give Eric his clone supply. If we couldn't provide Eric with enough clones to keep his pipeline filled, he would be free to fill it with random clones. But the priority was to be on map clones. That middle ground seemed to be okay with everyone. I went home to tell Mark O'Mara and John McPherson, who were leading our mapping efforts, of the commitments I'd made on their behalf. And not long after that, the job got harder still because John was unable to persuade any of his Sanger colleagues to give Eric any clones. There was not a good relationship. So we would have to build not half the map, but the whole map. Amazingly, over the next 2 years, Mark O'Mara's team fingerprinted or analyzed more than 350,000 clones, big 150 KB, 150,000 bases each. And John McPherson's group anchored those to chromosomes, so we knew where they all lay. The throughput grew to more than 10,000 clones a week. We calculated that Marco used 63 liters of agarose. Ladina Hillier used that emerging map to provide clones to Eric and to our lab and to others as well. And it was challenging initially because the map wasn't put together very far, and Ladina had to work really hard. But later we found out that despite the promises made in December and our exhaustive efforts to supply Eric with clones, he had apparently, with behind the scenes approval from Francis, gone ahead and put 2,000 random clones into his pipeline before anything else, before any of the clones we sent him. Because they were random, no one knew of them, and they ended up largely being redundant in the final assembly. The map came together amazingly well. It provided the background for both the public and the private assemblies that were announced at the White House. It had under 1,000 gaps by the fall of 2000 in a 3 billion bases. Through David Hausler at UCSC, the HGP recruited an amazingly talented graduate student, Jim Kent, to put these 30,000 clone sequences together into a whole genome. Because of repeats and problems in the data quality, he kept failing. Ladina and Marco, on the right there, Ladina, realized that the map could provide the necessary constraints to make the assembly work. With that, with these constraints, Jim produced a working draft sequence of the genome just days before the press announcement. Celera found their own ways to associate the sequence with the map, but the map enabled them to order their fragments and anchor their sequence to the chromosomes. The map also provided the means to get a high-quality sequence over the next couple of years. After 2 years of acrimonious exchange between the public and private sector that I won't detail, it's kind of fun though. Pressure from President Clinton eventually got our two sides together to agree to a truce. And part of that was the joint announcement of the respective draft sequences. That was Craig on the left and Francis on the right at the announcement. And simultaneous publication of our papers. Because June 26th was an open date on both Clinton and Blair's calendar. That was the date for the announcement. It had nothing to do with how we were sequencing. Totally arbitrary. But nonetheless, it was quite the celebration. There we are, Eric and Richard Gibbs, me and Rick, along with Jim Watson, celebrating outside after the announcement. Okay, so I'm running out of time here. I'm over time, but I'm going to finish anyway. The announcement didn't mean we were finished. We had to write up our story and coordinate enough with Celera to get everything published at the same time. Exploring what the genome was over those next months was exhilarating. And there's lots to tell, but I won't do it here. When the papers came out, we had the chance to finally look at the Celera sequence, and to our immense relief, even with using our sequence and our map in combination with their own data, because ours, after all, was out there in the public domain for anybody to use, their result was not much different from ours. And they did have the 100,000 gaps that Maynard predicted at that congressional hearing. We did indeed finish the genome, and 2 years later, we finished it again. NHGRI is very good at organizing press conferences. This was on April 2003 to coincide with the discovery of the 50th anniversary of the discovery of the double helix, again, an arbitrary date. We didn't have the whole sequence. There were actually pieces of it that were too full of repeats for us to do, and we'd have to wait until a couple of years ago to get a full sequence of the human genome. But it was very good, and it's fueled human genetics research ever since. So before I close, though, I want to spend a little bit of time talking about how the public project became committed to free and open data release, especially for those of you in the audience who take it for granted that sequence is just out there for everyone to use. When the pilot phase started in 1996, human gene hunters would often keep everything secret until forced to release data upon publication. And even then, they would reveal only as much as they had to. Many of the mapping centers were founded by gene hunters, seemingly hoping to use the genome funds to track down their favorite gene. John and I came from a very different ethos. In the 1980s, the worm community was small, with almost everyone having a connection to Sydney. There was a Worm Breeders Gazette that published one-page updates from what was going on in everybody's lab pre-publication. And there was actually very good adherence to not taking advantage of that information unfairly. It was that community that John was working with on the physical map, when he would post the latest on the physical map well before any publication and ask people to use it to find their favorite gene, and when they did, to give that information back to him. And with that kind of precedent, we followed that same kind of thing in sequencing, posting our sequence as soon as we got it. And when the internet became available, we put it out daily. So we came to that first Bermuda meeting in 1996, knowing that if the Human Genome Sequencing Centers were to become gene hunting efforts, that would make for competition, not the collaboration necessary to get the job done. And further, if we were seen by the wider community to take these enormous resources we were being granted to unfairly give us a leg up on finding disease genes, we would undermine community support. We proposed two things to deal with this. First, we would make raw data available. John was up there scrolling on this white sheet on the easel as the discussion was going on. I was leading it. We'd make raw data available to everyone as soon as we assembled it, so everyone would have an equal shot. It would also make obvious what we were sequencing to other groups. The second part was to forego any patenting of genes. That would eliminate monetary incentives, so that everybody would have an equal chance to explore the human genome and make the most of it for everybody. To my surprise and delight, people endorsed the ideas. And eventually this scrawl became redone, and these formed the basis of what became known as the Bermuda Principles. They're now well known outside the field of genomics, I gather, serving for a model in a diverse array of sciences. But they were the cornerstone of the public project. They were at the core of the dispute with Celera. We were under considerable pressure to collaborate with Celera because it potentially could get the job done faster and more cheaply. At one meeting, however, to explore collaboration, Tony White, the CEO of PerkinElmer, insisted that any combined product, anything that we worked on together, would only be available through the Celera website for some years to come, hopefully 5, he thought, and they would be the only source for any analysis, including any annotations that came from that effort. And we would not be able to use that sequence or any of that information to go on to finish the genome. In the end, we did not compromise. So the working draft sequence and the finished sequence are indeed out there for anyone to use however they want. Did you see that? Yeah. Oh, this is an article that's about the Bermuda meetings and how it came to be. And we also got Clinton and Blair to make a proclamation in March of 2000, commending us for our release of raw data and saying that DNA sequence should be made freely available to scientists everywhere. We would be in a very different world if not for the congressman from Michigan and a few other tipping points. It's been an amazing ride, going from a time when many opposed the genome, I was an outcast, to a time where getting a genome sequence is almost the first thing you do if you want to study an organism. Genomics has become foundational across all of biology. It's slowly changing the medical practice, maybe not as fast as we would have hoped, but it's having its impact. But with such a complex code, we still have much to learn. Good luck. Thank you. That's enough. We have time for questions? I went over.
Yes, we do.
Feel free to leave if you want to.
I wanted to ask you this for years.
Uh-oh.
Right. Um, in retrospect, was having Solera in the mix a net negative, irrelevant, or a net positive? I realize that in terms of the workload and the stress, it was obviously a negative, but in terms of actually getting the project complete, what was it? Not
So-
what could it have been, but what was it?
Yeah. It clearly got Francis to push for more funds faster than he might have otherwise. I don't know about that. The Wellcome was already on board. They were going to do their third of the genome, and that might have been enough to spur Francis. In terms of the actual work, we had to do shotgun early and shotgun late. That made it more difficult and more wasteful. I don't know. It might have pushed us a little faster, but I think that with the advances in technology that were there, I think it wasn't very much different. I don't think it was much faster or much slower than would have been, except maybe for the issue of funding.
And if Craig Venter didn't have access to all your data, how many holes would they have had then?
So they actually had in their paper one column where they reported what they had done with their data and what they did with our data. And then they never talked about what they did with their data alone. And without our map, those were random bits. They didn't know, unless they could use markers to place some of them, but if there wasn't big enough, I mean, it would have been a very different story. So.
We've got one online from Mitchell Vulgar who says, was this talk a preview for a coming book? Please.
No. Jay has a question in the back. I am writing a memoir for the advances in genetics and genomics.
You can include the answer to this in your memoir. So so like if you look back at like how you probably thought about things in 2001 and just what you thought the future would be like over the next 20 years, how did you think this would play out? What aspect of how you thought this would play out has been the most surprising in terms of the consequences and the uses and all of that? And then the corollary, what's been the most disappointing?
Well,
that makes
I
sense.
alluded to that at the end. I mean, I think I did not anticipate that genomics would just take over, that people really don't want to study an organism if they don't know what the DNA is. And it's become and I didn't anticipate that the sequencing costs would go down by 1,000,000-fold or whatever it's incredible that you can sequence a genome so cheaply. We spent, I don't know, $1 billion or something like that trying to get the human sequence. It depends on what you count and all that sort of stuff. And today you can do it for $10. I don't know. It's astounding. The disappointing thing, I was not as optimistic about the genome having an impact on medicine as people like Francis were. I thought it was just too complex. But I did hope that it would do more. When I go to my GP, he says, what would I do with the genome? Still today. I mean, it's clearly having an impact in healthcare. It's just been slower and harder to achieve than I had hoped.
Related question from Joe Felsenstein online. When I heard the phrase into the fire, I assumed you were going to deal with people's unrealistic expectations of what immediate health benefits would be. Is there another bigger fire?
It felt like I was in the fire at that time, Joe. I was under the gun.
Other question?
Oh, there's a,
Ultimately, what proportion of the human genome sequence came from Lander's lab?
um, in the final sequence, about in the low 20%, 30%, about 30% came from the Sanger, not quite a third. We put in about a quarter, about 25%. We had slightly more. than
Everybody
Lander.
else put together. So
Lander had more shotgun than any.
we had a huge amount more shotgun.
Not a
But
huge amount.
actual usable, oh, not, but actual usable sequence was about 20%.
At the final, in the finished product, it was about 20%. Low 20s, I believe.
So you alluded to this a little bit, that there are some people who are skeptical about the benefits of the human genome. And you said yourself, if I go to my doctor, he doesn't do anything with my genome. But there are a lot of people who were very skeptical about the benefits. And I guess initially, what would you say to those people now? What would you point to and be like, this is why this is really important?
To me, as a biologist, I view biology as enormously complex. Maynard, as a chemist, viewed it as quicksand. And he viewed the genome as a concrete block that kept you from sinking any farther. And I think it's that kind of, I don't know, that you can grasp all the information. I mean, it's all there in those 3 billion bases. It's the instruction book of how to make a human. We can't do it, but it's there. And I think that's a profound change in my view, to be able to have that information out there. In terms of practical benefits for medicine, I mean, the Mendelian diseases are mostly have their genes identified now. So people can understand what they are and try to devise solutions to them. It's clearly having an impact on cancer. It's not what people hope, but it's clearly having benefits. It's changing the way medicine thinks about disease. Does that? Okay.
One of the perennial questions, Bob, about the Human Genome Project is, where did you source the DNA? Who did you sequence?
There's I'll answer and then I'll tell you something else. There are too many stories that are coming to mind. Anyway, the bottom line is that 70% of the sequence came from an anonymous donor in Buffalo, a male and we can tell by his sequence that he was partly African American. But other than that, we don't know who he is. One story is that I was at a meeting. No, I won't tell this one. Anyway, apparently in the consent form, the donor was told that his sequence would not be used for more than 10% of the genome sequence. And when we were scaling up, we only had one library in 1996. And 1998, in the fall, a second library came, but that was already too late for the map. So we barged ahead. Fortunately, we didn't barge ahead without consulting the IRB at Buffalo in 1996, well before we launched the sequencing. And the IRB thought it would not be of enough importance to worry about. So we had that. A reporter, Ashley Smart, contacted me about 3 months ago with this information. And he's just posted the story in the Undark magazine from MIT Journalism School about this. I haven't even had a chance to read it. I just got the email today. So I don't know what he's gonna tell. He's might burn us for this. But anyway, it was one person for 70% of the genome. No, he was, there was an ad in Buffalo News Media recruiting donors. He was one of 10 anonymously selected donors whose blood was collected, totally wiped out of any identifiers, and passed on to Peter DeYoung, who just happened to pick that one tube to make the first library. He didn't know anything about any of it. He just picked one of 10. And the idea was that he would make 10 different libraries, but it's just too technically demanding to make that many clones from that many sources.
Another online from Cindy. What modern day project or question do you think needs a collaboration as massive as the Human Genome Project to advance science or biology?
I view the Genome Project as sort of a one-off. It was because it's a defined thing. It's 3 billion bases. We knew what the goal was exactly. And that's very hard to achieve in any other part of biology. It sort of goes back to this cement at the bottom of the quicksand. The rest of biology is too messy. And you know, there have been big projects like ENCODE and so forth. They've been successful, but they've also struggled because the goals are not, the endpoints are squishy. We don't know, we still don't know what all the genes are. We know they're 20,000 plus or minus. So I think, I don't know. If I thought hard enough, I could think of places I'd spend money. I'm not sure that the collaborations would necessarily be the best way to do it. And we were fortunate. It was, you know, we had John and I were the anchor, I would say. And it wasn't a huge group overall. There was a question back there.
Whatever happened to the private company? Did they go under or?
This was part of the .com. I mean, yeah, the .com boom. Their stock price went up from like $25 or $12 to $275. With one announcement, it dropped by $100 or something like that, because people thought they might be collaborating with us. That led to a congressional hearing in which I had to testify, in which we were blamed for dropping the stock market in one day by $60 billion or something like that. But yeah, Craig left after a couple of years, and it doesn't exist as far as I know. Does it? Does anybody know if it exists? It's a non-entity.
What did you mean, partly African-American for the donor in Buffalo?
We can tell by the variation in his genome that he's partly of African descent, but not entirely.
Like President Obama? Like,
Yeah.
Oh, okay.
Yeah.
Fascinating.
Yeah, like President Obama. Yeah.
I wasn't alive back then, so I wouldn't know. But I've my best understanding is that there was a lot of moral controversy surrounding the Human Genome Project at the time. How much would you say this controversy impacted your work, or did it impact it at all?
I mean, the controversy, the moral part was this part about data release. The sequencing part was this matter of scientific strategy and so forth. But I don't know, it made for a very stressful 2 years. I said we arrived at a consensus on how to proceed. That was very difficult to arrive at. I skipped over various meetings where people were shouting at each other, not shooting each other. about exactly what we should be doing. The press, we had Solera and Craig were very good at press announcements that put us in very poor light. He announced in January of 1999 that they had sequenced 95% of the human genome. What he meant was that they had random shotgun that covered 99% or 95% of the genome with our data. So they only had 80%, but with our data, they pushed it up to 90%. And those were just in little tiny pieces, most of them, and not very much use. But they went on to say that they had 97% of the genes. What they meant is that they could get a little tiny bit of a gene sequence and they didn't have the whole gene by any means, they had a little tiny bit. But the press took this up and said, oh, Solera's got 95% of the genome in January, they're done. It was just, every day there was something. In addition to having to scale what we were doing very fast. I mean, the map was done in a year and a half, 360,000 clones. In contrast, the yeast genome took, I don't know, 4,000 clones over 7 years. The worm map ended up with 7,000, 8,000 clones over 5 years. I mean, they did 350,000 clones in a year and a half. Oh, there was some pressure.
Yes, one last question. Good question.
So just completely out of curiosity, I know like how you mentioned for the Human Genome Project, especially in the beginning, I guess like the single sequencing method is mostly used, but now at least from what I've learned in class, like there are like, I guess, better and more advanced methods that are able to sequence like more base pairs at a time. So like say if something more advanced was like available to like the Human Genome Project at a time, like how would that technology change the way like the scientists kind of approach this and the funding structures and all of it?
That's a good question. You know, I said we could get 500 bases. To sequence the genome, we needed 50 million of those reads. And that was about right. Today, and we were working with clones for the human genome of about 150,000. Today, you can get sequence reads of over 100,000, even 150,000. So you don't have to go through that cloning step. You don't have to go through bacteria. And assembly is better because you don't have to do it 500 bases at a time. You have these huge base pieces, and it's that technology that has allowed others to get through all these repeats, to get the centromeres and the telomeres and all these other very hard to get sequences. So if, you know, and it's so much cheaper and easier. So if, you know, if any one of those pieces of technology was available in 1990, I don't think anybody would have worried about the human genome. There wouldn't have been any opposition. It was going to be too easy, too apparent. But as I said, when we started sequencing the worm genome, there was only 20 million bases of sequence from any source. So doing 100 was a little ridiculous.
Okay, we'll do one last question. I know we're a half an hour over, but I appreciate this. It's such a wonderful discussion. I kind of want to go for another hour, but.
I know you said that there will never be another Human Genome Project, but if I were to hold your feet to the fire, do you want to make any predictions about what the next, you know, big frontiers are, the next questions that need solved?
I mean, to me, the big question, the first big question is actually being able to read the genome. Right now, if I gave you the genome sequence of a chimpanzee and a genome sequence of the human, and you tried to read it just de novo without referring to knowing where the genes came from and so forth, where the sequence came from, you couldn't tell me whether that was making a chimpanzee or a human. We don't understand we don't understand that enough depth to read it, to figure out how it actually does what it does. So that people are working on that. Our department is a leader in that. We're doing better than anybody at trying to figure that out. But clearly the next challenge is to be able to write. And people are doing little bits and pieces. David Baker is writing new proteins. You know, and I don't know, I think the challenge is to be able to write a code that would make an organism. I think that would be awesome power, scary power. And something that would maybe be too powerful. But anyway, I think that's I mean, we have to be able to learn to rewrite the genome. And that's going to be a long time coming.
On that awesome.
That's why I wish you good luck.